Thanks, Bob, and hi, everyone. So as Bob mentioned, I'll be talking about two species in particular, so Rhodomyrtus robessens and Rhodomyrtus psidioides, which are two of the critically endangered species in New South Wales. Before I start though, I'd also like to acknowledge the traditional owners of the land that I'm presenting on today, so the Gadigal people, and also wanted to acknowledge other First Nations people who have shared knowledge of the species we're working on for this project, but also other projects on the North Coast, and in particular AJ and Tilly and our team, who are very knowledgeable and great at sharing that information. I wanted to mention two of our project partners. There are a lot of project partners involved in the project, but RBG Sydney in particular, we work closely with all of their teams, the RESA team, the Plant Bank team, their Living Collections team, and some of that work's been presented on already. And also the Australian Network for Plant Conservation, who have filled a real important gap in some of that coordination of the work, and in particular Amelia's work on the Living Collection Dispersal Project. And a special mention to Bob, Jeff and Angus. Setting up the project originally really relied on their expertise, and they were very free and willing to give that expertise, and that's continued over the years since, so it's really appreciated. So just quickly a bit of background on Saving Our Species. It's a program that's been mentioned a few times. It's the New South Wales Government's flagship threatened species program. Across the first 10 years, which takes us up to 2026, has been 175 million allocated to it. It has a species-specific approach, which has its pros and cons. It uses a prioritisation process to select projects, and also really focuses on consulting with experts and building partnerships. And I think this project is a good example of that. So in New South Wales, we currently have 4 species that are listed as critically endangered because of declines from myrtle rust. And all of those are funded under SOS to some degree, and I'll talk about the 2 species that I've identified today. So Rhodia Robessens, it's been mentioned quite a lot. It's a medium-sized tree, grows up to about 25 metres tall. It's very widespread. It is distributed from the south coast all the way up into southeast Queensland. It's really common across a lot of different vegetation communities as well. It's a great pioneer species. That was something that Christy mentioned the other day. And now it's declined so rapidly that it's listed as critically endangered in New South Wales, Queensland, and also at a Commonwealth level. So native guava is very similar as well. It's a small tree, gets to about 12 metres tall. It's also quite widespread. It's distributed from just north of Sydney at Gosford up into southeast Queensland. It's another great pioneer species. It grows, seen quite commonly in littoral rainforest, but also you see it in other rainforest and wet sclerophyll communities. It's also a species that was quite often used in land care plantings because it was such a easy to grow, quick growing species. So one thing that's common to those 2 species I'm talking about, but also the other all 4 listed species in New South Wales, is that they're having a really rapid decline, and that decline is solely because of the impact of myrtle rust. So flowering and seed production's affected. For most of these species, seedling recruitment is basically non-existent, and even trying to get viable seed is something that is not very common at all. And we know that the cohort of plants that we see out there in the wild at the moment are likely to be the last natural recruits of this species, and when they decline, then that's the species gone. So because that rate of decline has been so rapid, we know we're losing individual plants, we know we're losing entire populations in some situations, and with that loss we have a loss of genetic diversity. And we also know that genetic variation that is out there is really important for the long-term recovery of these species. So our initial emergency response is really focused on trying to capture that genetic diversity and understand it better, and that was done primarily through germplasm collections and leaf sample collections for the conservation genomic work. So we've focused on 4 main areas over the last 4 years. One is to undertake rapid field assessments. So initially, I suppose we knew that these species were declining rapidly at the sites we'd looked, but we didn't really know if that was going to be uniform across their entire distribution. So we've focused on looking at that impact across the entire distribution and recording things like infection levels. We've also completed a lot of those leaf sample collections, and those genomic studies have been presented on already, and trying to get the ex situ collections established. So I'll just quickly mention one of the tools we're using. Because we're collecting lots of different types of information, we're collecting leaf samples for the genetics team, we're collecting germplasm collections. We wanted some way that we could tile that together with what we're observing in the field, and we've been using a free program called Epicollect. And it's a really easy program for developing basically a form builder that you then download onto your phone or your tablet, and you collect the information in the field. It means that when we have sent that information down to the genetics team or the living collections team, they've got access to what we've observed in the field as well, and it ties all of those 3 areas together. So I'll just start to talk about what we've seen in the field for a couple of these species. Rhodomyrtus robessens. Like I said, it's normally considered to be a tree that grows up to 25 metres tall. Here we just have a height class distribution for it. What we're really finding now is that most plants are in this 2 to 6 metre tall range. While we do still see larger trees up to 20 metres, and some of those do have a pretty good level of leaf cover, it's quite uncommon really, and we've done extensive surveys for this species. Rhodomyrtus psidioides is a very different situation. It's really reduced to suckers now. So you can see on the screen there are lots of small plants that look like seedlings. They're actually suckers that have come off the roots of what would have been once an adult plant. And you can see in the other image next to it where that plant has actually come off that root system. So most of these plants are under 50 centimetres tall. Majority are probably under 30 centimetres tall, and they come up and die back on a cycle every 1 to 2 years. So one other thing I'll mention with this species, we have found a few larger plants that have looked healthy, and I think the teams in Queensland have found a couple that look promising. But really, in terms of across all the sites we're looking at, it's very rare to find a larger plant that's got any level of leaf cover on it still. So I'll just sort of talk about some of the germplasm collections. So the real focus was to try and increase the number of plants and the number of lineages we had represented in the collections at Mount Annan. So prior to 2018, I think there were only 4 lineages of Rhodia Robessens down at Mount Annan. Since then, through the project and collections of other people, we've increased that to 35 different genotypes now, representing 21 populations, and the work that Veronica's done down at Mount Annan, we now have 283 individual plants in that collection. For Rhodomyrtus psidioides, it's a similar situation. Prior to 2018, there were only 2 lineages represented. We've now got 30 populations and 60 different genotypes represented, and a massive 570 individual plants. So like Amelia was saying, these ex situ collections can be quite large and take a lot to maintain and manage. So I won't really spend a lot of time talking about the conservation genomic work. That was presented by Stephanie, and Jason has also touched on aspects of it. But I did want to say that it's been a really important tool for us to have managing this project. It's given us a really good idea of what's happening in the wild in relation to genetic diversity in the species. It's helped inform us and helped us understand whether or not our ex situ collections capture that variation. And it's also has been and will be a very important tool when we look at selecting groups of plants for different purposes. So that might be cryopreservation, it might be dispersal of mini collections, and eventually even things like seed orchard arrangements and dispersal of these plants. So in terms of future priorities, we really want to continue looking for some of the potentially resistant individuals of these species. We want to expand our assessments to include other species that we know are rapidly declining. So the 3 on the screen here are species that have been identified in Queensland as declining rapidly, and we've also seen the same impact on the north coast of New South Wales. And that'll give us a better understanding of what community level impacts are happening because of myrtle rust. And we're able to do this through the key threatening process, which is also an avenue for working on myrtle rust in New South Wales. And finally, just in terms of our future priorities, I think the ultimate goal or success for a project like this is to get these plants back out into the wild. While the ex situ collections are a really important and essential part of that process, essentially in the long run, we want to have the plants back out in the wild. We didn't start this project to just have collections in pots. So that's something we've just started working on with the Botanic Gardens in New South Wales. And it's something we're hoping to progress over the next few years with RBG Sydney. I think the other thing that we'd like to improve going forward as well is how we engage with First Nation people on this project. The initial emergency response was very much a frantic collection of germplasm material as quickly as possible. Now that we're moving into the next stage, and it's a longer term stage that may hopefully eventually result in plants going back out into the wild, we really need to improve that engagement with First Nations people. So yeah, that's all I wanted to cover today. Thanks for listening.
Thanks for that. Just a quick technical question. Have you observed Rhodamnia suckering?
Yeah, Rhodamnia does sucker. We've seen small suckers, but we've also had collections at some sites where we've had multiple plants in an area of 5 to 10 metres that may have been larger, and they were all clonal as well. So that came out through the genetic work. Not as common probably as Rhodomyrtus.
Thanks.
Other questions? Yeah.
Nice talk, Craig. This is perhaps a question for later, but just following on from what Richard was talking about with white pine 2 days ago now, or yesterday. Neither of these plants are sexy or iconic, and they don't look like a little fluffy bear.
I think that's your perspective, Angus. But
How do you see trying to get momentum of funding through the type of stuff that Richard was talking about with some of these species?
Yeah, I think one thing that's really important when we consider funding is we need to have some runs on the board as we progress this work. So have something to say that, you know, this is feasible, this is what we've done so far, and you know, use that to continue to get funding to support this work, because we know it's going to be expensive work and it's going to be very long term. So I think that's one way we can try and combat that, and take lots of nice photos of them.
Rob?
Thanks very much, Craig. That's really great work that you guys are doing here. I'm just wondering, I'm surprised that you don't have any climatic risk information to support the work that you're doing. I'm just wondering, you know, what sort of priority you think that should have, and suggest that, you know, Bureau of Meteorology could certainly supply the data that you'd need to, you know, run a risk model and use that.
Yeah, I think then two aspects to that. In terms of the myrtle rust suitability zone. These plants occur totally within that myrtle rust zone on the east coast. But when we get to that point of potentially translocating resistant lineages back into the wild, then looking at climate suitability is probably something we should be doing as part of that. I know it's something we do with other species in our team.